Good morning, everyone. Captain Sauceda again, and today we're going to be exploring the Duffy blood group system. A system that's not just important in transfusion medicine, but also deeply tied to human evolution, infection resistance, and even genetics. So what makes Duffy really special is that it bridges those disciplines. It is a blood group system, yes, but it's also a chemokine receptor, a malaria resistance marker, and fun fact, the Duffy gene, which is located on chromosome 1, just like Rh that I taught the other day, was the first human gene ever assigned to a specific chromosome. So that's a huge milestone in genetics, and it actually laid the foundation for mapping of the entire human genome. So while Duffy might not be getting as much attention as ABO or Rh, it has an incredible scientific legacy and a powerful clinical impact. So today we'll walk through those antigens, antibodies, the enzyme behavior, and more. And by the end, I hope you'll see just how relevant and fascinating this system truly is. So let's start off with a quick timeline of the Duffy blood group system. In 1950, the system was named after a patient with the last name of Duffy. This patient was a multiply transfused hemophiliac, and they developed an antibody against this unknown antigen. And since anti-D was already in use, they already used that for naming, the new antigen was designated Duffy, or FY. 5 years later, Sanger reported that the majority of African Americans tested were FY A negative, B negative, meaning they lacked both the Duffy A and Duffy B genes. This null phenotype is referred to as FY and has clinical and evolutionary implications. Then later on in 1975, it was discovered that red blood cells lacking Duffy A and Duffy B were resistant to infection by Plasmodium vivax and Plasmodium knowelski, which are two parasites that are responsible for malaria. So some biochemistry on the Duffy antigen. The antigen is a multipass glycoprotein that spans the red cell membrane 7 times. Similar to Rh that we discussed the other day that spanned it 12 times. So in addition to the transmembrane regions, it has 3 extracellular loops, which are important for its interaction with those chemokines and those pathogens like the Plasmodium. The gene that encodes for the Duffy antigen is called ACKR1, which stands for atypical chemokine receptor 1, and this protein acts as a non-signaling or a scavenger chemokine receptor. So playing roles in inflammation and immune modulation beyond its role in transfusion medicine. The Duffy A and Duffy B antigens differ by only a single amino acid at position 42. Duffy A has glycine and Duffy B has aspartic acid. This diagram shows that the predicted structure of the Duffy protein, it highlights the position of both Duffy A and Duffy B epitopes and their locations in those extracellular regions. So as far as genetics, the Duffy gene, as we discussed earlier, is ACKR1. It was formerly known as DARK. It's on chromosome 1, and the Duffy locus is syntenic to the Rh locus, so that means that it's on the same chromosome, but far enough apart that the linkage cannot be demonstrated. So that it does segregate independently. There are a total of 6 blood group systems on chromosome 1, so that's a significant amount. So there are 4 main codominant alleles. You have Duffy A, Duffy B, FY, and FYX. Duffy A and Duffy B are the 2 most common alleles. The FY is a silent allele, which results in no expression on the RBCs, though there could be some expression on other tissues. And this is one that's very common in individuals of African American descent. And Duffy X is a rare allele that is a weakly expressed variant. And we'll talk about each of these a little bit more in detail. So while there are 4 basic alleles, these give rise to 6 antigens. Well, really only 5 since Duffy 4 is obsolete. So you can see here that Duffy A and Duffy B are also called FY1 and FY2. But the other ones, FY3 is not called Duffy C. It's just called FY3 from that point on. And just a point of clarification in case you're confused at this point. So an allele is a version of a gene or DNA, and that determines the instructions for making a specific protein or a specific feature. But an antigen is a protein or a marker that's found on the surface of a red cell that can trigger an immune response. And sometimes an allele and an antigen have the same name. So while in this case, Duffy A or Duffy B or FY1, FY2 are both alleles and antigens, they each have other antigens that fall underneath them. So on this chart, you're going to see some antigen frequencies. Notice that they are numbering them 1 to 6 in this case. So Duffy A, they have as FY1, Duffy B as FY2. But just for me to stay consistent, I'm going to continue to call FYA and FYB those things and not call them FY1 and FY2. But notice that there is no FYC again. It's FY3, 4, 5, and 6. Duffy A is present in about 67% of white individuals, but only 10% of black. And this disparity is largely due to the high prevalence of the null FY allele, FY in people of African descent. And Duffy B is more common than Duffy A in both of the groups. FY3 is expressed in those that have either FYA or FYB. So it's nearly universal in the white population. You see here it's greater than 99.9%, but only found in 32% of those black individuals. FY4 is considered rare in the white population, but it's found in 98% of the black population. So this antigen is thought to be expressed only in the absence of FYA and FYB. So the presence correlates with the FYA negative, B negative phenotype. And 5 and 6 you can see on there the same story as FY3, the same percentages. And we'll go over those individually. So now that we've looked at how each individual antigen appears in those different populations, we'll go ahead and shift to looking at the phenotypes. So of course, meaning the actual combinations of those antigens on the red cells. So Duffy A positive, B negative is the most common among Chinese individuals with over 90%. Duffy A positive, B positive is a codominant expression, and it is most common phenotype among whites, but not very common among anyone else. FYA negative, B positive is present in about a third of whites and 22% of African Americans. FYA negative, B negative is the null phenotype and is it's especially significant. So it's very rare in whites and literally not seen in the Chinese population, but it's in almost 70% of African Americans. And this is due to the high prevalence of the FY silent allele, which leads to no Duffy antigen expression on those RBCs. This offers protection against Plasmodium vivax. So Duffy A and Duffy B are the two most common alleles. They're antithetical antigens and are very well developed at birth. An individual can express one, the other, both, or neither. One important thing to note about Duffy is they don't store well in a liquid or in a frozen state. The antigens just don't tend to survive. So if you need to do some testing and you don't have a fresh sample, it's always good to request a new sample because you will not have a good chance to appreciate that antigen-antibody interaction unless it's in a fresh sample. So the antibodies to the Duffy A and Duffy B antigens are mostly IgG. They can bind complement, and although they're capable of crossing the placenta, they're not a common cause of HDFN. Anti-Duffy A is much more common than anti-Duffy B. As a matter of fact, it's 20 times more than Duffy B, and it's also much more immunogenic. Anti-Duffy A and anti-Duffy B tend to show dosage, meaning they react more strongly when a person has 2 copies of that target antigen, meaning they're homozygous. However, they may not react as strongly as some of those other systems like KID. These antibodies can cause delayed hemolytic reactions. And one of the challenges is that the titers may drop below a detectable level over time. So the antibody may not show up in pre-transfusion testing, but it can still cause a reaction. And it can also take multiple exposures to these antigens before they actually start to make antibodies. So anti-Duffy A and Duffy B are often found alongside other antibodies, especially those of the Rh system and the Kell blood group systems, in patients that have received multiple transfusions. So because they've received those multiple transfusions, they're often exposed to a lot more highly immunogenic Rh and Kell antigens, and Duffy kind of just tags along. It's kind of rare to find the Duffy antibody in a specimen all by itself. So if you ever happen to notice that, it could be a good sign to just keep on looking. It's just not common to find it by itself. So FY3 is a unique antigen in the Duffy blood group system. There are other blood groups that follow a similar pattern with the third antigen of the blood group. So I think yesterday you heard about a couple of those. So this should be kind of familiar to you. FY3 is not encoded by a separate allele. Instead, it's a structural or conformational epitope that's present when either Duffy A or Duffy B is already expressed on the cell. So if a person has either antigen, they will also have FY3. So the only ones that do not have FY3 are those that have the FY A negative, B negative phenotype. So those FY individuals of African descent also don't have FY3. So the chart here just shows you exactly what I just talked about. So the only time you won't have FY3 is when you don't have A and or B, or A and B. Anti-FY3 is a rare antibody that's detected against a shared structural component of both FYA and FYB antigens. So because of that, it's not possible to separate that anti-FY3 from either anti-FYA or anti-FYB in serological testing. So just if you can make the correction on your sheets, that first bullet point, one is supposed to be anti-FYA and the second one is anti-FYB, like you see on the screen. I think your printout probably has B on both of them. So if you can just go ahead and fix that part. So it is often associated with mild hemolytic transfusion reactions and even mild cases of HDFN. And it is clinically significant, but it's just typically not very severe. So in white individuals, anti-FY3 will usually react with the Duffy positive cord blood cells because most of those do express FYA and or FYB. In black individuals, that anti-FY3 does not react with cord blood that is Duffy positive. And this may be due to the fact that some black donors carry that GATA mutation that silences antigen expression on the red cells, but not in those other tissues. So it is just important to note for when you're studying the differences, right? So this question was on our exam, and you could maybe see it on the certification exam. So again, whites, they do react with Duffy positive cord blood, but in blacks, it does not react with Duffy positive cord blood. So this slide answers a really interesting question. Why don't we see more anti-FY3, especially in those people with that FY A negative, B negative phenotype? You know, considering that 67% of the black population have this phenotype, you know, it's just a little unusual to be negative for an antigen, but then also not to produce that antibody when the body is exposed to it. Most donor blood expressed either has FYA or FYB, which means then they also have FY3. So when someone who is FYA negative, B negative receives that transfusion, they're pretty much all the time going to be exposed to FY3. So the key here lies in that before mentioned GATA mutation. So that's of course found in many individuals of African descent. And this mutation essentially silences the Duffy gene expression, but only on the RBCs. So we call that an erythroid null phenotype. So the Duffy gene still functions normally on the tissue cells. So on those tissue cells, it continues to produce FYB and therefore also produce FY3 antigens. And since they're already being expressed by the tissues, the immune system recognizes them as self, not as foreign, and therefore it doesn't produce antibodies against them when they're exposed through transfusion. Those people may still, however, form an anti-FYA, since those are truly foreign, as only FYB and FY3 are on those tissues. So this slide highlights the differences between what a person inherits genetically, so the genotype, versus what is actually detected on the red cells, which is the phenotype. So, I mean, I'll start off with saying that, you know, FY4, Duffy4 is obsolete. So this is what is thought is happening with that antigen. They thought that the antigen FY4 produced that FY, or that null allele, the silent allele, but what they found out is FY does not produce any detectable antigen on those RBCs. So when someone inherits that FY from both parents, they will have the FY A negative, B negative phenotype, just meaning there's no Duffy antigens present at all. So FY4 was once believed to be a distinct antigen produced by that, FY, but it's not actually present on FY A positive, B positive, or any Duffy positive phenotypes at all. Instead, FY4, if it was there, was mostly associated with individuals who were Duffy negative. So here's where knowing the race of the patient kind of helps. So you notice the phenotype for the first two is both FY A negative, B positive, but the genotypes are different. The white population is most likely to be homozygous for FYB, so they have FYB, but the black population is most likely that they'll probably have FYB, FY. So knowing that, what the race of a patient is can make a huge difference in how you triage those antibodies that are produced. So if you're working something up and you see something like this happening and you know the race, you're going to kind of try to go down one route, kind of saving you some time in figuring out what it is. So the key thing just to remember here is that two different genotypes can lead to the same phenotype. So anti-FY4 is a bit of a historical mystery in the Duffy blood group system. It doesn't exist. FY4 doesn't exist. So if FY4 were real, it would be expressed only on the red cells of black individuals with Duffy negative or partial epitopes, and hence the antibody would only react with those. So it's theorized, not to be present on FYA positive, B positive RBCs of any white Duffy positive phenotypes due to their lack of having that FY. But the pattern of reactivity doesn't align with the known serological principles, and anti-FY4 was difficult to isolate and reproduce anyway. So eventually it was determined that the reactivity, once thought to represent anti-FY4, was likely due to antibodies against other Duffy-related epitopes or just non-specific reactions altogether. So as of today, FY4 antigen and the antibody are no longer considered valid or clinically relevant, and they were essentially retired in the ISBT system. So it's more of a footnote than an actual antigen, but it's still important to understand that just in case you have a question on the certification exam that asks about Duffy4. So because multiple genes can give you the same phenotype, it can lead to a pretty difficult pedigree. So FYA positive, B negative can be either FYA, homozygous, or they can be FYA, FY, which is heterozygous. And this is due to the fact that it's two different genes for a trait. Duffy A negative, B positive can either be homozygous for FYB, so FYB, or heterozygous, meaning FYB, FY. So knowing the race could kind of help you narrow this down. If they were right, they're most likely going to be FYB. Now, if you're FYA positive, B positive, or FYA negative, B negative, you will be homozygous for those respective genes. So, how does this look in a pedigree? We can take a look at that. So here we have a family. We have 2 parents and 3 children. It's always most difficult to do a pedigree using the phenotype, so that's why we're going to go ahead and do it. So first, you're always going to assume that everything is normal. So based on that, which child here doesn't appear to fit? So just take a look at it real quick, and you see the first child on the left is FYA positive, B negative. So they could get their FYA from the parent on the right, and they're B negative, so that's fine because they must have inherited that from the parent on the left. The child in the middle got their FYA positive and B positive, so they probably got their A positive from the parent on the left and their B positive from the parent on the right. But then what about that last child? So let's take a look at that last child. They're FYA negative, B positive. So with the way that this looks, it looks like it should be FYB, FYB. So, but where did that second FYB come from, right? You only see that the parent on the right has one FYB, but the other parent doesn't have any. So how do you explain something like this? Well, I will show you. So in this case, that first parent, the parent on the left, isn't FYA, FYA at all. They're actually FYA, FY. So the child is not FYB, FYB. They're FYB, FY. So because of the rare allele, it makes it possible that this person is the parent and, maybe not end up on Jerry Springer for this. So FY5 is a unique antigen in the Duffy system because its expression depends not only on the Duffy gene, but also on the Rh gene. This is a result of the interaction between the two. If you recall, both Duffy and Rh are on chromosome 1. FY5 is not present on RBCs from individuals who are FYA negative, B negative, of those that are black. It's also absent on any RBCs that are Rh null, regardless of their Duffy phenotype. And that means if Rh antigens are missing entirely, then Duffy 5 will also not be expressed. And that's something to know. FY5 is expressed on RBCs from white individuals with that same FYA negative, B negative phenotype. I think when Bill gave us this lecture, he told us there were just a handful of white people that have had this phenotype. So very few that they've even had a chance to research. So this just kind of highlights the fact that the phenotype alone does not predict an antigen expression, and those genetic backgrounds and interactions really do matter. So I can get into why this happens, but just briefly, it's not super important at this level, but essentially it has to do with whites lacking that GATA mutation, so that ACKR1 gene is still transcriptionally active in the RBCs. So this means that other parts of that Duffy protein are still present, including those structures that form FY5. So FY5 is detectable on these, whereas in blacks, the GATA1 promoter mutation prevents expression of that Duffy protein on those red cells entirely. So no Duffy protein means there is no structure for that FY5 to form, even if the gene is present in that tissue. So that's a little bit different. So it's just important to know here that FY5 is not present on the FYA negative, B negatives of blacks, but it would be present on the RBCs of whites with that same phenotype because of that GATA mutation in the black population. That's a test that I know we were asked on our exam, so that's just something that I would know because that is kind of a unique situation. So with all that, so we can deduce that anti-FY5 will react with the white FYA negative, B negative, but not that black FYA negative, B negative. So anti-FY5 will react with those white FYA negative, B negative RBCs because the Duffy gene is still transcribed on those red cells, even when FYA and FYB aren't expressed. So FY5 can still form. However, that does not react with RBCs from the black individuals that had the exact same phenotype because, again, of that GATA mutation. So anti-FY5 is associated with the delayed hemolytic transfusion reaction, but not with HDFN. This is because that Duffy antigen, it just doesn't tend to provoke a strong fetal response. Okay, and the last antigen is FY6. So this Duffy antigen is recognized by murine or mouse-derived monoclonal antibody. It's present on all red blood cells, except, again, those from individuals with the Duffy A negative, B negative phenotype. But it is present on Rh null cells. So that's one of the key differences between FY6 and the other Duffy antigens like FY5. So FY6 serves as a critical point of entry for that Plasmodium vivax. So initially, scientists believed that the parasite used Duffy A or Duffy B to gain entry, but we now know it's actually FY6 that facilitates that process. So if someone is either FYA or has either FYA or FYB, they're also going to express that FY6, so they are susceptible. Now, here's where it gets a little bit more evolutionary. So in parts of Africa where P. vivax were prevalent, the individuals with the FYA or FYB positive were more likely to die from that infection. So over time, with natural selection, it favored those who were FYA negative, B negative, and therefore they lacked that FY6. And that's why today, that 67% of the people of African descent are homozygous for the silent allele, FY, and therefore they're resistant to P-Vivax. And there's a little graphic there just showing you that. So anti-FY6 is a murine monoclonal antibody, meaning it's produced in mice and recognizes the FY6 epitope on the Duffy glycoprotein. So just important to note that there is no naturally occurring human antibodies to FY6. So we don't see any hemolytic disease of the fetus in newborn or transfusion reaction because it's just not in people. And that's because the immune system in humans just doesn't typically respond to FY6 as a foreign antigen. And I'm sure there's probably reasons for that just haven't been super researched. So his antibodies primarily in research and diagnostic testing. So particularly to help differentiate the FYA negative, B negative phenotypes or other Duffy variants. But again, it's all based on a laboratory model. So there's no evidence that humans naturally make any kind of anti-FY6. So FYX is essentially a weakened form of the FYB antigen, and it's caused by mutations in that Duffy gene. So the mutations result in reduced expression of that FYB antigen on those red cells, which can sometimes be missed during standard serologic testing. The FYX is found in about 13% of white individuals, and while it doesn't produce its own unique antigen, it can still react with some anti-FYB reagents, though the reactions are usually weak or inconsistent. So one key feature, so red cells with that FYX phenotype can absorb or adsorb and elute anti-FYB, which just further confirms that some level of FYB expression is present. It's just at reduced levels. So the only thing you really have to remember about FYX is what I have in bold and larger font there. FYX just basically equals a weak FYB. So if you understand FYB, then this one will be easy. It's just a weaker form of that. All the other serological results will be the same, but weaker. So when we're testing for blood group antigens, it's important for us to understand how they react with the enzymes. Some antigens are enhanced or destroyed when the red cells are treated with the proteolytic enzymes like physin, papain, bromelain, chymotrypsin, and pronase. So the Duffy antigens on this chart, FYA, FYB, and FY6 are destroyed by the enzymes. So this means that if you treat those red cells with the enzymes and no reaction occurs, it's a good indication that the presence of a Duffy antigen has been denatured. So it's probably Duffy A, Duffy B, or FY6. So in contrast, FY3, 4, and 5 are not destroyed by enzymes. So it's kind of easy to remember because they're all in a row, 3, 4, and 5 are not destroyed. And this is the way that it can help you differentiate between those various Duffy antigens and those complex serologic workups. So just one enzyme to note on this slide is trypsin. So it generally does not destroy Duffy antigens, so it's not used diagnostically in the same way as those others. And as you can see on the table, the enzyme treatment can enhance some antigens like RH, KID, and Lewis, but it's going to destroy others like MNS and Duffy. So if you understand these patterns, it can help you, and it's a key in interpreting antibody panels and resolving those discrepancies that you might find. So ACKR1, we talked about this earlier, but it's also known as the atypical chemokine receptor 1. It's the gene that encodes the Duffy antigen receptor. So you might see it referred to by its older name, DARC, which stands for the Duffy antigen receptor for chemokines. So this primary role for this is chemokine regulation. So it binds and removes excess inflammatory chemokines like interleukin 8. This helps to maintain immune homeostasis. So interestingly, it's also been shown to bind unwanted chemokines from prostate cancer cells, which has implications in cancer biology and immunology as well. So this is where the connection is to transfusion science. So Individuals that are FYA negative, B negative due to that GATA mutation and, lack Duffy expression on those red cells, but they still can express ACKR1 in those tissues. However, this loss in RBCs can impair that chemokine clearance. So the studies suggest that the Black populations with that FYA negative, B negative phenotype may actually have a higher incidence of prostate cancer, possibly linked to altered immune regulation. So you're at a lower risk for malaria, but at a higher risk for prostate cancer. But if I can put my old cytology hat back on, prostate cancer will get every man if they live long enough. It's just one of those cancers. So it's just interesting to note here, just a higher incidence in the Black population. And it makes me wonder if maybe do they live longer? And maybe that's why we're also seeing it. So it's just interesting for me to think about that as well. So in conclusion, Duffy isn't just a red cell antigen. So it also plays a key role in that immune surveillance, inflammation, and even cancer biology. So it's not a boring blood system at all. It's actually one of the more interesting ones, even though it's not quite as large or confusing as some of the others.